Hello guys, I have studied about microbial carcinogenesis and I find this very interesting. So, EBV is a gamma herpes viridae family DNA virus in which there are two viruses present, HHV8 which causes Kaposi sarcoma, and this we have is EBV. So EBV is associated with many B cell lymphomas, and how it is happening, that is what we are going to see. So EBV binds to the CD21 receptor present in the B cells. So by expression of some proteins, this causes proliferation. And as we know, prolonged proliferation results in accumulation of some mutations that results in cancer. So, but this proliferation is normally controlled by our cytotoxic T cell, which will destroy them because the affected proliferating B cells will express the EBV proteins. So, they are getting destroyed by the T cells. So, this is associated with some cancers like Burkitt's lymphoma, right? So Burkitt's lymphoma is associated with endemic areas of malaria. So when malaria is common, T cell immunity will be low. So there is defective clearance of these proliferating B cells because some of them may suppress their antigenic expression, so they will survive, and that results in Burkitt's lymphoma. Now, we can also say that in severe immunodeficiency like AIDS or post-transplantation conditions, there will be mutation, there will be T cell lowering. So when we affected with EBV, there is no one to destroy the B cell from proliferating and eventually mutation. So what happens is that they need not to worry about the T cells as in Burkitt's lymphoma. So will so they will express the antigenic protein and they don't have to worry about the T cell because it is not there. Then there is nasopharyngeal carcinoma because EBV usually causes pharyngitis. But here there is not T cell affected, but the PD L1, L2 ligand expression by the nasopharyngeal carcinoma cells, that is a B cell carcinoma cells, uh, will express these proteins that will suppress the T cells. So that results in proliferation and mutations. You know. Then there is this mutated B cells in Hodgkin's lymphoma by IgH rearrangement and somatic hypermutation. The next step is the occurrence of increased NF-kappa beta expression, which may be EBV associated. That is why we are including here. That may not be EBV associated. Then followed by mutation, and then Hodgkin lymphoma, which is Reed-Steinberg cells. So I have mentioned only the antigenic cells. So what are the antigens that are produced? LMP1 and EVNA2 are the major antigens. So LMP1 is responsible for LF kappa beta increase because it produces an activity similar to that of CD40, and EBNA2 is not signaling. So especially we're talking about this CD40. So CD40 is a co-stimulatory molecule um, that is present in the B cell, and when it expresses the antigen via MHC2 to the T helper cells, and the CD40 ligand will attach to the CD40 in the B cells, and it will produce the cytokines that will cause the proliferation of the B cells like IL-2, and also it can change the immunoglobulin from IgM to IgG, IgE, or many subtypes, depending upon the interleukin released by the T cell. But now here you see it is not happening because we are independently by LMP1 causes proliferation, causing proliferation of the B cell, and that results in occurrence of mutation. So the concept is simple. EBV is affecting the B cell, that results in proliferation. When it is not controlled by T cell, that results in occurrence of mutation, eventually cancer. This is the main part. This is the main part of the this concept. All the other are related thing you can easily relate. Thank you.